Good evening, everybody. We're glad that you are here. I'm Adejoke Ayola, the Dean for the School of Health, and I'm excited about this presentation and this opportunity for us to actually share with you part of a partnership with Michigan State University. So I will start by introducing one of our partners, and then he will introduce our speaker for today. And that's Dr. Mark Brevey. Yeah, so he actually graduated from Calvin in 2004. He graduated from political science, and he also has a minor in business. Yeah, and Mark Brevey is the director of the Community and Government Relations at Michigan State University, and that's the College of Human Medicine. His duties include developing relationships with business and community leaders, as well as university officials, in order to promote and encourage visibility for the college through coordinated events. And we're having one of such events here. And we've been in partnership with MSU for so many years, and we're glad that Dr. Brevey is here again today. And like I said, he's a graduate of Calvin, and he lives in Grand Rapids here with his wife, Emily, and their children, Jonah and Micah. Please let's welcome Dr. Brevey.
Well, thank you, Dr. Ayola. That is a bit of an upgrade. I do not have my doctorate, but I am a very proud Calvin grad. Yes, it's such a pleasure each year to come back to Calvin. We want to thank Calvin University for hosting the event in this great facility. This facility is near and dear to my heart because my wife and I actually got married right here. So again, it's always great to be back at Calvin. This lecture would not be possible without the co-sponsors of the event, Calvin University and Michigan State University College of Human Medicine. This is the 10th year, by my count, of the Your Health Lecture Series event at Calvin University. We hold this event due to the unique partnership we have through the Early Assurance Program agreement between our institutions. Calvin University students are eligible for early admission to medical school through this program. Now, without further ado, I would like to introduce our speaker for this evening. Maggie Caulfield received her PhD in immunology from Northwestern University in 2013 and completed postdoctoral training in neurology at the Mayo Clinic in 2016. Dr. Caulfield then spent 3 years as an assistant professor of biology at Calvin University, teaching molecular biology and neuroscience, as well as studying sex differences in immune cell responses in CNS autoimmune disease. Dr. Caulfield has been a senior postdoctoral scholar in the Steece Collier Lab since September 2021, where she focuses on the preclinical analysis of gene therapy-based interventions to reduce common therapeutic side effects in Parkinson's disease. And the Steece Collier Lab is actually here in Grand Rapids at the MSU Grand Rapids Research Center, downtown Grand Rapids. Her laboratory skill set includes cell, molecular, and histological techniques such as cell culture, flow cytometry, RNA in situ hybridization, qPCR, Western blot, ELISA, immunohistochemistry, and confocal microscopy. I knew I should have just cut all of that before coming up here. But when she isn't in the lab, she enjoys spending time with her kids, cooking, and sailing on the many local lakes with her family. And I will just say thank you all for coming. Obviously, we have a lot of snow here in Grand Rapids, first big snowfall. The roads have not been great, but we have a great turnout in person, and we also have people joining us online. So those who are joining online, at the end of the talk, we will have the ability to take questions online as well. So put that in the Q&A. Without further ado, Dr. Caulfield.
Thanks, Mark. We set you up for that one with the business degree and all the science terms. That was a tough one. Sorry for that. Thanks so much for coming today. I'm going to talk to you a bit about Parkinson's disease and about transplant therapies in Parkinson's disease. So the focus of the talk is really thinking about neuroregenerative therapies. So things that essentially regrow neurons to help Parkinson's patients. I'm going to give you an outline first of what I'm going to cover so you have some idea of what's coming. We're going to do a lot in kind of a short amount of time, so I hope I can take you along with me. First, just an overview of Parkinson's, a bit about the disease, where in the brain is affected, the pathology and the symptoms. I'm going to talk about a drug called Levodopa, which is commonly used for most Parkinson's patients. And I'm going to talk about a side effect of this drug called levodopa-induced dyskinesias. And I'm going to show you what our rat model looks like. Then I'm going to move on a bit to some clinical cases describing neurotransplant for Parkinson's patients. I'm going to give you a really good clinical case, and I'm going to show you a bad and ugly clinical case. So you start to see how this has progressed throughout history and where we're going into the future. I'm going to introduce you to a molecule called brain-derived neurotrophic factor, which we're interested in the context of transplant, and particularly a SNP, so a single nucleotide polymorphism in BDNF, in the BDNF gene, that affects the way grafting works in people who carry that allele. And the very end, I'm going to tiny touch on how this all plays a role into personalized medicine currently and into the future for Parkinson's patients. If anyone has a family member or friend that has Parkinson's, or you've watched someone that has it, you're well aware that it's a relentless, progressive neurodegenerative disease. About 1 million people in the US have Parkinson's currently, and 60,000 people are diagnosed in the US every year. This leads to a really large economic burden on the United States, estimated about $52 billion per year. And for individuals, the medications typically cost around $2,500 a year, and surgical therapies would be around $100,000 per person. Unfortunately, the incidence of Parkinson's disease is predicted to double in the next 25 years. And we can already see this trend happening. This is largely due to a combination of factors. One is our coming into contact with more Can you hear me now? All right, thank you. I'll try to be less erratic with my movements. Part of this increase in incidence is us coming into contact with all these industrial byproducts. So herbicides, pesticides, all the stuff in the water, microplastics, all those things. The other is people are living longer. Parkinson's is a disease of aging, so the longer you live, the more incidence there's going to be. A kind of interesting counterintuitive thing is that actually people who smoke have a lower risk of Parkinson's disease. So as less and less people smoke, that also increases the number of people that we're seeing with Parkinson's disease into the future. The vast majority of Parkinson's cases are sporadic. So they come from a place that we don't know exactly what's causing them. Generally, we think that it's a combination of factors. So your family history, your genetics, and environmental factors. Like I mentioned, pesticides and herbicides exposures, toxins, living in a rural area, etc. These things are combining, it's thought, to give rise to these types of cases. There are also a small percentage of familial Parkinson's cases. So these are your genetic predispositions or genetic mutations that are causing Parkinson's. These are usually associated with young or early onset Parkinson's, and they tend to be quite aggressive. So here comes Anatomy 101. I should have BB up here doing this. I was thinking about him the whole time, like, I'm going to mess this up, and then BB's going to be like, she's wrong. So there's two areas of the brain that we're particularly interested in tonight. One is part of the basal ganglia. So the basal ganglia sits inside the forebrain, underneath the outer part of the brain. And this basal ganglia is important for motor control. That's what most people think about. Also motor learning, behaviors, emotions. And there's a particular part of the basal ganglia that we're interested in, which is called the striatum. In humans, the striatum is made up of two parts, the caudate and putamen. So you're going to see those words on my slides a lot. So just think caudate, putamen, striatum. Those are what we're interested in. And I'll get to why it's important in a minute. The other part that's important for Parkinson's disease, besides the basal ganglia, is the substantia nigra. So this is found basically dorsal a bit to the basal ganglia, and this is the area that's affected, particularly in Parkinson's disease. These two areas, so let me orient you. Here's the substantia nigra in this image. Here's your striatum with your caudate and your putamen. They talk to each other via a dopamine pathway. Sometimes it's called the nigrostriatal pathway. So it's really kind of crazy to think about, but the dopamine-producing neuronal cell bodies live in the nigra, down here, and their dendrites extend all the way up here, and their terminals are found in the striatum, and that's where the dopamine is released. It looks something like this. This is a different view of the same idea. This is actually a schematic of a rat brain instead. But here you see in the nigra, there's the cell body of the neuron, and its axon, and then its dendrites come all the way up to the striatum and release the dopamine. And here the dopamine is blue. In Parkinson's, there is pathology in the nigra. And so in a normal, healthy individual, this is a slice, a brain slice from the nigra. You see this dark line where these arrows are pointing. Kind of cool thing about dopamine neurons in the nigra, they also make pigment. And so to the naked eye, you can see these dark cells in the striatum of a healthy individual. And if you section the nigra and you do histology and stain them, in this image, the happy, healthy dopamine neurons are purple. So you can see there's lots of big, fat, happy neurons. In a Parkinson's patient, they lose those dopamine neurons in the nigra, or they don't function very well anymore. And so you can see, actually, the loss of that black pigment suggests that those cells are no longer making pigment, they're no longer making dopamine, or maybe dead. You can see it in the histology. There's very few big, happy, fat dopamine cells. And the pathology behind this is really thought to be a consequence of aggregated proteins in the cells in the nigra. These aggregations are called Lewy bodies. So sometimes you hear like Lewy body pathology associated with Parkinson's. And a lot of it is this protein called alpha-synuclein. And it aggregates up, it gunks all up, and it causes stress to the cell, and the cells dysfunction or die. And that's the basic level what's happening in Parkinson's. We just don't know why that's happening necessarily. So here is our nigra, again, talking to our striatum. And in Parkinson's, when we lose those dopamine-producing cells, we lose this connection, and there's no dopamine, or very little dopamine, being produced in the striatum. If you don't know what dopamine is, you're probably wondering, why do we care about dopamine? She said it like 1,000 times. This is why. So dopamine released in the striatum is responsible for your ability to move, essentially. And so when you have a decrease in that dopamine, you have motor symptoms. This is our typical old white dude Parkinson's patient. If you Google Parkinson's, you're gonna see something like this. And they have this very classical stoop posture, shuffling gait, tremors at rest. They have a masked face, and these are all motor complications. It's become accepted more recently that there's also a whole lot of non-motor dysfunction in Parkinson's disease, some of which likely predate any of the motor complications, particularly sleep disorders and GI issues. So constipation is really common in early Parkinson's. And so many people think if we could catch some of these non-motor symptoms, that might be a window for your therapy that we haven't really appreciated before. This graph looks complicated, but it's not terrible. I'll walk you through it. This shows the relationship between, if I can get this pointer to come up so the Zoom people can see me, the relationship between the loss of dopaminergic neurons, here in blue, over time. So this would be like over a 20-year time course or more. And in the red, the motor dysfunction. So we're starting to lose dopamine neurons as we get older. Parkinson's is starting to set in, and the motor issues start to come up. And once they start to be pronounced enough that someone knows something's wrong, they go into the clinic, and they're diagnosed with Parkinson's. It takes some time for this to happen, and you've lost a whole lot of dopamine neurons by the time this happens. Sort of relatedly, but with a different kinetics, is the non-motor. So a lot of these non-motor symptoms come up earlier, before the motor symptoms, and then they have a slightly different trajectory over time. Sort of luckily for Parkinson's patients, there's a drug called Levodopa. Levodopa is considered the gold standard for Parkinson's disease patients. Levodopa is a dopamine precursor, and it is actually transferred across the blood-brain barrier. It's taken orally, generally, and it is trafficked across and then converted into dopamine and then released from the neurons. So it's essentially a dopamine replacement. And this graph is showing from a particular paper, the lower the bar is, the more negative, the better the motor score is. So the better the levodopa is working. Levodopa works really well for a lot of people. It doesn't always work for everyone, but it is definitely the first line of therapy for Parkinson's patients. Unfortunately, in about 50% of Parkinson's patients, by 3 to 5 years after diagnosis and treatment with levodopa, they start to get this side effect called levodopa-induced dyskinesias. As opposed to the sort of rigid lack of motor motion that you think of for Parkinson's, a dyskinesia is sort of like a hyperkinetic motion. It's uncontrolled, unwanted, involuntary motion. And it can be quite debilitating. Here you see a series of pictures of an individual with Parkinson's disease, and she actually is spilling out her cup and dropping her cup because she's moving so uncontrollably. She can't sit up straight, and she can't hold a cup of water. After 10 years of having levodopa therapy, about 90% of Parkinson's patients suffer some degree of dyskinesia. So we consider this an unmet clinical need in Parkinson's. Michael J. Fox, probably one of the most famous celebrities with Parkinson's disease. He was a really early onset Parkinson's patient. I want you to watch the way he moves. We're just going to watch him for 30 seconds. It's not rigid. It's very, very fluid, and he's moving a lot in sort of abnormal ways. He has levodopa-induced dyskinesias. Maybe. If I can do it.
What did you know about Parkinson's?
Somebody's grandmother had it. I mean, it was not a thing that I noticed or thought about. It didn't enter into my world. I was a 29-year-old guy.
Did the doctor know who you were?
Yeah, and in fact, that's one of the few times in my life I felt like saying, do you know who I am? It's ridiculous. You can't tell me that. This was a case when I just thought, it's preposterous that this is happening to me.
His slurred speech, muscle stiffness.
So you can appreciate when he talks, you see how his head is moving. You can tell there's some sort of vocal issues. He doesn't talk totally clearly, but all of that head motion and body movement, that's the dyskinesia. That's not the Parkinson's. We can model this in rats, and we do. So first I'm going to show you a rat who has levodopa, and it is motorically enhanced. How do I get my arrow back? Here we go. So this is a really happy rat on a pretty high dose of levodopa, which means he has a whole lot of dopamine right now. So he's sniffing and he's all excited, and this is a happy rat. So again, I'll play him one more time. See how he moves, sniffing, grooming. Now I'm going to show you a rat that has levodopa-induced dyskinesia. This rat looks very different. So see how his body is twisted, his hind limb is stuck out, his right forepaw is clenched, and he does a repeated motion with his right hand. I'll play it again. So this is how we're modeling that symptom in rats for potential therapies to get rid of dyskinesias in humans. So you could think, okay, Parkinson's disease, we don't have enough dopamine. When we give them a dopamine precursor back, you get these terrible dyskinesias. So skip the drug, let's just put the dopamine-producing cells back in to the brain. This was a really exciting idea in the 70s and 80s and 90s. Basically, to orient you again to the areas that we're interested in, you have the substantia nigra down at the bottom with the dopamine neurons. And so this is just the striatum and the nigra kind of pulled out of the brain. And you have the dopamine neurons and then them extending up into the striatum. Parkinson's disease, again, those nigral neurons are degenerated, they're dying. There's very few connections to the striatum. So let's do some neuronal grafting, and would this cure Parkinson's? So you would put in actually the dopamine-producing cells into the striatum, where you need the dopamine. They don't have to grow those really long extensions from the nigra. They can just be where they're needed, replace the dopamine, fix the Parkinson's, no levodopa needed. Sort of interestingly, historically, the cells that have been used for these transplants come from fetal tissue. So these can be miscarried, stillbirths, ectopic pregnancies, and elective abortions. And it's because the cells that are accessible in those, the fetal tissue, are precursors. So they can grow into dopamine cells in the striatum, and that's the source that had been used historically. They put these, you might wonder how you get these cells deep into your brain. You use what's called a stereotactic apparatus. So the person's head or the rat's head sits in a frame. It has measurements in 3 dimensions, X, Y, and Z. You have certain coordinates that you put it in, so then you get the needle right into the exact spot that you want it to be, and then you can inject the cells using a mechanic injection, essentially. So I'm going to show you 2 clinical cases. One, like I said, has a good outcome, and one not so great. These both happened in the 90s and have been published about quite a bit. So this is a 55-year-old man who had Parkinson's. This is a PET scan, so the more red or the more white, the more dopamine there is. And so what you see glowing is his striatum, so you can see he really doesn't have much dopamine, particularly on this one side. They did a unilateral transplant, so they just put the dopamine neurons on one side. You can see that side makes a whole lot of dopamine 1 and 3 years out. This worked really well for this patient. They had an almost near loss of their Parkinson's symptoms, and when they were still on levodopa, it lasted longer. So they could take a dose, they'd have a lot of benefit from it, and it would last a long time. And they were doing so well that they actually took them off levodopa. So that's up to 3 years after transplant. What happened between years 3 and year 10? Eventually the symptoms got severe enough, the Parkinson's symptoms, that they did introduce levodopa. And this patient stayed stable for about a decade, which is pretty phenomenal. And they didn't have any notable or troublesome dyskinesias. And they saw a neurologist every 6 to 9 months for the next 14 years. So what happened in those 14 years? By year 14, the severity of the Parkinson's did increase. The levodopa became less effective. Some cognitive impairment ensued. 18 years post-transplant, the effect of the transplant was gone. It was no longer helping this patient. And this patient died 24 years after transplant of cardiac insufficiency. They were 83 years old at that time. After this patient passed away, researchers and clinicians looked at his brain, and here are the striatum from the non-grafted side and the grafted side. The brown equates to dopamine production. So where he didn't have a graft, there's no dopamine. Where he does have a graft, there's a whole lot of brown. And that means those dopamine neurons survived for 24 years in this patient, and they are real happy. So you can see the blow-up version, big fat cell bodies, lots of axons and dendrites coming from them. They're producing a lot of dopamine. So this was really an important finding, that these cells lasted for so long in this patient, and they got benefit for over a decade. I mean, to go from being severely Parkinsonian to having no motor symptoms is pretty astronomical. Some of the later issues this patient had were due to white matter atrophy. So the cognitive dysfunction is due to the shrinkage of the white matter in their brain. And here is a slice of their cortex, and the ventricles are really big. And that tells you that that part of the brain is shrinking, and so that causes the dementia. They also still had Lewy body and alpha-synuclein pathology in their nigra. So over time, that continued to accumulate, and so eventually the Parkinson's kind of won out in the end. So that's a pretty good case scenario. It might not seem that good, but it's pretty good. Now I'm going to show you a bad and ugly case. So this is actually a 59-year-old woman. These are her PET scans before her transplant, a year after, and 2 years after. She had a bilateral graft. So she has grafting on both sides, and you can see the red on both sides. She survived for 16 years, and she also had a long, extensive survival of her graft and innervation of those neurons for 16 years. She had 0 clinical benefit from this graft. Her Parkinsonism got worse over time. She had severe graft-induced dyskinesias. So this isn't from the levodopa. This is from the graft itself, actually caused that unwanted motion. And she was so sick that 8 years after she got grafting, she also got deep brain stimulation, which is another sort of surgical intervention for severe Parkinson's patients. So you can tell sort of how desperate she was for care. A couple other clinical trials were done, or larger studies were done in the early 2000s. One was published in the New England Journal of Medicine in 2001. In their cohort, they saw grafting benefited their younger Parkinson's patients, but not their older ones. And 15% of their patients had graft-induced dyskinesias. Another study from 2003, none of their 23 patients showed any benefit from the grafting, and half of them had graft-induced dyskinesias. There's also been a long-standing ethical and political concern about using fetal tissue for transplants, as you can imagine. So in 2003, there was a worldwide moratorium on fetal transplantation for individuals with Parkinson's disease. This isn't a law. This is a thing that researchers and clinicians agreed that we need to take a step back. This is not working particularly well, and we're actually making some people worse. We're giving them a whole different issue, the graft-induced dyskinesias. The question is, why doesn't this work every time? Works sometimes, but why not every time? Well, there's a whole lot of reasons. The way the embryos are collected, dissected, the procedure using for that differs from lab to lab. The way the tissue is cultured or matured ex vivo can vary. The surgical procedures vary. The patients that are selected vary, as is the post-operative care can be different. The immunosuppressives they're on after grafting and how much more pathology they accumulate over time can all differ in all these patients, making it incredibly complicated to have a one-size-fits-all therapy. Clinical trials using transplantation with fetal tissue restarted about 4 or 5 years later, and the newer studies all take into consideration host age, disease severity, and a whole lot of other aspects. Our lab is particularly interested in the genetics behind what makes a patient a good, suitable patient for grafting and what doesn't. So in our lab, we're particularly interested in a molecule called brain-derived neurotrophic factor, BDNF. And this looks really scary as a graph, but what's really important is when BDNF works in the brain, it causes neuronal survival, plasticity, and differentiation. And these are all good things when you want to regrow cells or grow cells in the brain in Parkinson's disease. Within the BDNF gene, there is a very common SNP, so a very common single nucleotide polymorphism. And basically what this means that within the prodomain region of the gene, in some people, they have a substitution where they end up with a valine, or a methionine instead of a valine. 20% of the world's population carries this allele, and in some populations, in some areas like Japan, it's up to 70% of the population has this substitution. BDNF has been associated with a number of neurodegenerative diseases, particularly a decrease in BDNF. So we see less BDNF in Parkinson's disease, Alzheimer's, Huntington's, depressive disorders, and schizophrenia. And the SNP that we're interested in, the RS6265, has also been associated with some of the disease aspects in those different diseases, but we don't know a ton about what it's doing. What we do know is that it decreases the amount of BDNF that's available in the brain. So if you have this SNP, you make less BDNF. So BDNF modulates neuronal survival, differentiation, branching, and maturation. So we hypothesize that this common human SNP, the RS6265, if you have it, it may explain why some people are refractory to transplantation and or develop graft-induced dyskinesia in humans. So we wanted to test this. To do this, we wanted to look at the role of this SNP in both the host and the donor cells. So we did a little mix and match experiment. This was actually done by a grad student who's sitting right over there. This is part of her thesis work, so Carly was kind enough to let me show this data today. So we mixed and matched the host and the graft. So we had wild-type hosts. We had hosts that were homozygous for the MET allele, the MET-METs. So they had the SNP, two alleles of the SNP. And then they had either wild-type or MET-MET grafts. So this is how the experiment worked. Made the rats Parkinsonian. We induced levodopa, induced dyskinesias. They either got a dopamine or a sham graft from one of the genotypes. These came from embryonic day 14 rat pups. We isolated the mesencephalon and made a single-cell suspension, and then we used a serotactic apparatus to put it back into the rat striatum. We continued the levodopa treatment, and then we tested for graft-induced dyskinesia. So, grafts from both genotypes into both hosts protected these animals from levodopa-induced dyskinesias. This is a good thing. So, this is wild-type hosts, wild-type brains. If they got a no graft, a sham graft, that's the gray line, they had bad levodopa-induced dyskinesias throughout the whole experiment. If they got wild-type or the MET grafts, they were protected. That's why the blue and black lines are lower post-grafting. They were protected from that dyskinesia. That's good. Same thing if they're a MET-MET host. It didn't matter the host or the graft genotype, they were protected from dyskinesia. What was particularly interesting was that when the rats were the MET-MET hosts, so the RS6265 hosts, that's going to be the green squares and the orange triangles, particularly when they had a wild-type graft, they were susceptible to graft-induced dyskinesias. So there's something about the way the wild-type cells connect to the MET cells in the striatum in a BDNF-deficient environment that causes this unwanted side effect of the graft. There are some animals who got the MET graft in the MET host that also had graft-induced dyskinesias. It just wasn't all of them, or the majority of them. If you have wild-type hosts, their graft-induced dyskinesias, regardless of if it's a wild-type graft or a MET graft, look just like the sham. So they're not having graft-induced dyskinesias. So what do we want to do next? Well, I told you that if you have this MET SNP, you have less BDNF. So let's put the BDNF in to a MET animal who's getting a wild-type graft and see if that protects them from GIDs. So if you were a MET, I'm actually a MET-MET. If you are MET-MET or MET, if you were getting a graft, you'd also need BDNF supplementation. That's the idea. We're going to do this winter. We use these osmotic mini pumps. They're made by a company called Alzette. The pump actually sits subcutaneously in the skin. There's a little tube, a little catheter that goes into a cannula that sticks into the brain, and we will release the BDNF right above the graft and hope that if there's enough BDNF in the environment, we'll protect the MET animals from graft-induced dyskinesia. This represents an appropriate supplementation for MET individuals with Parkinson's undergoing transplant, potentially. That's the idea. So in the field, what's going on now and what's going to happen in the future? The history of transplantation of Parkinson's has been varied. Where are we now? It doesn't matter that you can't read these words. You just need to look at the colors and the circles. These are all of the current clinical trials as of January 2022. And the top 3 are therapies that are disease modifying. So these are actually working on the mechanism underlying Parkinson's. The bottom 3, the bottom curve, are all symptomatic. So you could tell the majority of the drugs and therapies that are being tested for Parkinson's clinically are still just treating the symptoms and not the underlying disease mechanism. However, there are about 3 or 4 clinical trials in the brown, in the upper disease modification. Those are cell transplantation trials that are going on currently. The difference about most of these transplants is that the cells are no longer coming from fetal origin for the most part, but they're coming from the patient's actual cells. Their own fibroblasts are being isolated, pushed into a stem cell phenotype, and then pushed into a dopamine-producing neuron, and then replaced into the patient. What this cycle allows for is personalization of this type of therapy. So let's say you're a MET and you need a transplant. They take your fibroblasts. Well, those fibroblasts have this MET allele. Well, you could take those fibroblasts from that patient, culture them, fix the SNP, grow them appropriately, turn them into dopamine neurons, put them back into the person. So you don't have to worry about immune rejection. You are no longer taking tissue of fetal origin, and it's really personalized to that patient. And with that, that's the end. That's the big ta-da! It's gonna help, it's gonna work for everyone. I wanna thank our lab at Michigan State, our boss, Kathy Steves Collier. She's the brains behind these ideas for treating levodopa-induced dyskinesias. She's spent her entire career essentially working on this for Parkinson's patients. As I mentioned, Carly, our graduate student, this was her data. Those graphs I showed were her data, so thank you, Carly. Our lab manager, and then 2 really special Calvin alums are in the audience. You might, if you have been around a while, you might notice Molly Vanderwerp and Caleb Mathai, who are sitting in the middle here, are also working in the lab, and we're really thankful to have them as part of our team. And I would be happy to take any questions or comments. Thank you.
Thank you, Dr. Caulfield. This is just really interesting for a personal reason for me and just in general. 2 questions. The first one is, when you showed that cycle of the personalized approach, how long does that take? And then my related question to that is as you addressed at the beginning of the discussion, and several times throughout, the age of the client does matter. So with this particular approach, how does that factor in?
Those are great questions, Dawn. To your first question, I don't actually know the timeline for this sort of culturing. It's not something that I've been a part of clinically. It's just what's coming out in the field currently. So I don't have personal experience with that. frankly, and this sort of work's been done or is being done in some other groups too. When I was working at Mayo Clinic on multiple sclerosis, they were doing something similar because there's neurodegeneration in MS as well. And so this is not unique to PD, and primary cells from people or animals don't live terribly long generally because they sort of poop out, I don't know how to say it, after a while. So they have to do it relatively quickly. And I think some of the hang up for this and why it's taken a while to get it into the clinic is to get the specifications just perfect to grow these cells just right so they do what they're supposed to do and they don't grow too fast or become a different cell type or all the possibly bad things that could happen in response. So I think there's some caution there. But I do see this as there are some of these in clinical trials in mostly phase 1 at this point, phase 1 and phase 2. Your second point to the age is a really interesting one from a research standpoint. Most research is done, preclinical research is done on young animals. Bummer, because that's not what an old person is like. We have taken on a number of studies in aged animals. They are incredibly difficult, and their brains are definitely less susceptible to intervention. So you would think that probably translates to humans. A lot of the clinical trials in humans are likely going to try to focus, at least in the beginning, for safety profiles and small numbers on the younger PD patients in their 50s, and some of these patients who have genetic Parkinson's because they tend to just be younger. I think the hope is, as we learn more over time, if we can get it to work in a less aged brain, then we can optimize that for the aged brain. I think it's going to be a stepwise process to get there. Other questions, comments, concerns?
So I have one from online. It says, thank you for this discussion. Can you speak to whether these findings trials could be relevant in atypical Parkinsonian disorders like PSP and MSA that respond more poorly to Cinemet?
Well, that person clearly knows their clinical neurodegenerative disorders. I don't know the answer to that, and so I'm not going to speak to it because I don't feel comfortable speaking to that. I would say that generally, Parkinson's patients who don't respond to levodopa do move on to other therapies more quickly, and so I would consider that to be more likely for other diseases. I know some things like deep brain stimulation is really, really helpful in people who have something like essential tremor, which is a single sort of motor deficit that they can target very specifically with deep brain stimulation, which doesn't work quite as well in Parkinson's. So I think there are going to be individual therapies that are appropriate for different diseases and would have to be optimized for that particular pathology. From the Zoom.
What is the difference in the success rate of Parkinson's treatment using fetal stem cells compared to adult cells turned to stem cell?
Historically, the fetal stem cells has been the most successful, and that's why it was sort of carried through. I think the push to using these sort of D and re-derived pluripotent stem cells is relatively new, and as far as I know, none of these clinical trials are finished. And so there's not much data on this. So that's why I'm sort of pushing out this is like happening now and will be optimized into the future. But previously, the fetal grafts are what work the best in animals, and that's why that was first tried in humans.
So another question, when you showed us some of the slides that showed the Lewy bodies, I think you may have addressed this, but I either didn't quite catch it or didn't understand fully. In the successful transplantation trial, did Lewy bodies still grow and exist? Therefore, there really wasn't anything that could be taken as a connection between the transplantation of the cells and the development or advancement of Lewy bodies?
You're correct. So the transplant is happening in the striatum, and it's essentially working to replace the dopamine in that particular area. Lewy bodies can form in lots of areas in the brain over time, and in Parkinson's, most concerned about the substantia nigra, and that is progressive. And so if someone's predisposed to those aggregations, it's likely that will continue. So the one patient that did well for a decade, the neuronal replacement was enough to keep them stable, but eventually the amount of aggregation kind of overruled the transplant. Yeah, so it's not targeting that pathology. So there are many other people who are studying and working on not letting that aggregation happen. We're sort of post-disease diagnosis, trying to make the levodopa work better, essentially.
I'm curious, for a show of hands in the audience, just in general, if you had the opportunity to work on this level of research, how many of you would right now have an interest in it? Can you raise your hand? I'm just curious.
On. Caleb and Molly, you better raise your hands.
Like, I'm thinking a new day job here.
I take you, Terry, any day.
Well, thank you so much, Dr. Caulfield, for your presentation. It's extremely interesting research. I was wondering if there's any connection between the amount of time a patient's been taking levodopa prior to transplantation and the effectiveness of transplantation.
That's a good question, and I don't know the answer to it. I don't know. It's possible. So the longer you're on levodopa, generally the worse the levodopa-induced dyskinesias are, and we think that that's because of the way the cells sort of reconnect to each other. When you have the dopamine, the cells in the striatum start to make these weird connections to each other, and so the more miswired your striatum is, the worse off you're going to be just in general. However, when you put a transplant in, you're sort of replacing all that dopamine, so it might not be a huge deficit. I'm sure there's data on that. I'm just not familiar with all of it. Thank you. Get your steps in, Terry.
Are there specific toxins or categories of toxins that have been linked with increased instances of Parkinson's? And then also, do you see sex differences in treatment outcomes in your rat models?
Those are great questions. I wonder if someone's feeding these questions that I know. So the first one, the toxins, mostly pesticides and herbicides. So there is a correlation between rural living on a farm. There are these pockets of Parkinson's cases in these areas, so there's thought to be a correlation there. I don't know if there's a direct causation that's been shown yet, but there's definitely an understanding that this is detrimental. The other sort of interesting toxins that are being studied, and actually part of the reason why we're funded by the Department of Defense, is because chemical warfare agents used on our veterans in historical wars have also been linked to Parkinson's symptoms. And so the DoD is really interested in studying Parkinson's in veteran populations and their exposure to toxins while they're serving. Oh, what was the second one? The sex differences. In this study that I talked about for transplant, we only used male animals. In other therapies, we have a gene therapy that we study. We do think there are some differences between the way male rats and female rats respond. Male rats tend to be more uniform in their response, how their LIDs, or levodopa dyskinesias, are decreased with our gene therapy. Female rats have a more varied response, and we're sort of backtracking and trying to figure out what that's related to. It's likely related to hormonal environment, whether or not the female rats are still cycling the levels of estrogen and progesterone. And these are things that are really important to be studying in Parkinson's disease. Parkinson's is a 1.5 male to female ratio, so slightly more or more men get Parkinson's than women, but it's not like women don't get it. And basically to date, all of the trials have been done in men, Lots and lots of the preclinical work have been done in men. Obviously, men and women are not the same. And so the NIH, I don't know, maybe close to 10 years ago, instituted that you have to have a rationale for using only one sex in your studies, or you have to use both. And our boss and our lab thinks it's really important to be studying in males and females, so generally we include both sexes when we can and are starting to tease apart some of those differences.
I'm wondering how you think the new therapies will impact the financial burden on the U.S. as well as the financial burden on patients as this is growing.
Yeah, that's a really interesting question. We get really excited about these therapies and the science behind them, right? And that this really kind of advanced therapy could work. This sort of therapy would not be for everyone, right? You're going to have the right patient at the right time with the right genetics. So I don't really ever foresee something this invasive being a widespread treatment for Parkinson's. I think there's going to be, and I think the hope for personalized medicine is that it varies based on what's appropriate for you. So you don't just do the same thing to everyone, that we can start to decipher differences in the individuals with the disease and pick appropriate treatments. So I think this is always going to be a relatively small group of people who do this. It's similar, currently, deep brain stimulation. It only works kind of in a subset of people, and you don't start poking your brain until you really, really want to go there. So you have to be dedicated to that sort of therapy. So I think, but for the individuals that do it, it's going to be expensive. So we're talking about in the hundreds of thousands of dollars range. So it's not going to be something taken lightly.
Me again. So I guess the other thing that I've been thinking about listening to you talk tonight is the areas of medicine where now when we do genetic testing, we can predict increased risk factors for certain types of diseases. I'm just wondering if this research has the potential to inform further research that could give us the opportunity with genetic testing to identify risk factors for development of Parkinson's and open that door, kind of like what we've done with breast cancer is the example I'm familiar with.
Yeah, I think that that's a reasonable idea. I think for people with genetic Parkinson's, that's pretty clear. You know, if they're early onset, severe, test them for genetics, there's probably a familial PD. For PD, sporadic PD, maybe it'd have to be like a whole profile of genes and SNPs and things, right? So it's not going to be a single gene or two genes. It's probably going to be you have less BDNF. And there's actually multiple SNPs in the BDNF gene that is carried around the world. So maybe you have a combination of these SNPs. And you also have a different SNP and a different neurotrophic factor that causes it. And so when these combined, you're more predisposed to it. It'd be a complicated story, but where I think it might be more immediately applicable is people who have this BDNF SNP that we study are less likely to respond to levodopa therapy. So if you knew you had this SNP, you either maybe wouldn't start on levodopa, or you would be watched really carefully because you know that it's not going to last very long. So I think more immediately we could think about personalizing the therapies. Detection is a whole nother game, but I mean, it's possible, right? It's the hope that we could get there.
You mentioned that the MET SNP is far more prevalent in Japanese populations. Does that translate to higher risk for them, or is there something perhaps environmental that needs to interact here to really cause that to trigger?
I don't know the answer to that, Cooch. It's a great question.
I to come up with more research projects.
Google it. No, I think that's great. And actually, a lot of the clinical trials in the pluripotent stem cells are coming out of Japan. So as research groups, they know that it's more prevalent and that they need to be working on some of these transplantation models and stuff. So it's, I don't know. There's clearly a lot I don't know and a lot we don't know.
Interesting Kathy Steese-Collier just commented,
That's my boss.
RS6265 is not associated with PD incidents.
Is she saying that's interesting? I don't know what you're saying.
I don't.
Kathy, I don't know what you're saying. So basically, so if you look in the upper, under B, this SNP, this R6265, is only associated with the levodopa not working as well and the altered age of onset. So, but to Kuchi's point, if you had the SNP in combination with something else, it's not directly associated with incident to this point, but it's possible in combination with environmental or other genetic factors that are found in certain populations. You can see how that story could be strung together. I think that's what she's getting at.
She did write further, just therapeutic response to levodopa.
Yes.
Okay. I kind of jumped the queue to read Kathy's. So back to question. If I understood correctly, there's a substantial decline in dopamine long before symptoms emerge. Is there a way of detecting this?
Yes. So I'll just pop back. They're referring to that graph where you see the dopamine go down over time well before people end up in the clinic. If you took a dopamine PET scan with a fluorescent dopamine marker or dopamine transporter marker, you would see a decrease at a certain point once it's detectable, potentially before motor dysfunction. However, people don't routinely just take PET scans for no reason. They're expensive. There wouldn't be reason to take one, so it would be hard to justify doing that before you're sick for most people. If you have familiar Parkinson's disease, maybe that's more justified, just like people who have family history of breast cancer get mammograms earlier, maybe they would watch you a little bit more closely. So yes, it's possible, you just don't know who to scan necessarily.
We got another one online, last one online, and then we'll see if there's another one in the room. Is genetic Parkinson's dominant or recessive? And would it present like Huntington's disease where you would be unaware if you were passing it to your kids?
So it can be either. So there are different mutations. So, and they're both autosomal. So some are dominant and some are recessive. And I don't know if you would know if you passed it on to them. You probably wouldn't until you could test them. And you'd have to know, for some of them, like the recessive, your partner's genetics too, whether or not you should be concerned.
Do we have a final question in the room?
Oh, come on.
I had the mic for a long time, but so if both parents passed away from Parkinson's, their biological child then should have maybe an increased level of concern, or that still could be pretty sporadic that both a mom and a dad could have it.
Because it's fairly uncommon that it's actually genetically based, about 15% of cases, likely they both just got sporadic Parkinson's and you really aren't worried about passing it on. It's pretty common, especially if those parents were living a long time, into their 80s, 90s, and above. So it's really a disease of aging and a compounding factors, genetic environmental factors. So I probably wouldn't run out and get tested unless those people had early onset Parkinson's, severe, those sorts of things. That would tell me, okay, well, this might not just be sporadic. It's not this aging process. Something else is going on that's underlying this.
All right. Well, let's thank Dr. Caulfield one more time. And I just want to thank Dr. Ayula one more time and Terry Crum for the great hospitality here at Calvin. We just come to expect it every time Michigan State University College of Human Medicine and Calvin collaborate on this event. So With that, thank you all for coming and joining us online, and if you're driving home, drive safe. Thanks.